Welcome to G Fun Facts Online. Today, we are diving deep into one of the most profound questions of human existence. Why do we age? And can we stop it? For centuries, the secret to a long life was thought to be a simple lottery of fate, or perhaps a divine reward for a virtuous existence. But today, we stand on the precipice of a radical new understanding. Longevity is not merely a roll of the dice. It is a complex interplay of genetic architecture and environmental signaling, a code that science is finally beginning to decipher. In this episode, we are going to explore the intricate genetics of human longevity. We will dissect the heritability of lifespan, identify the specific genes that act as the guardians of our youth, analyze the biological clocks ticking within our cells, and look at the futuristic interventions that may one day allow us to rewrite the boundaries of human existence. Let's begin with the fundamental puzzle, the heritability of lifespan. For decades, the scientific consensus was somewhat discouraging if you were hoping for a genetic silver bullet. The belief was that the genetic contribution to human lifespan was relatively modest, somewhere between 15 and 30%. This estimate came largely from traditional twin studies and family genealogies. The prevailing wisdom was summarized by the famous adage, genetics loads the gun, but lifestyle pulls the trigger. In other words, your genes mattered, but your diet, exercise, and smoking habits mattered much more. However, recent research has dramatically upended this view. A landmark study analyzing thousands of twin pairs in Scandinavia, published in Science and other major journals, revealed a startling nuance that changes everything. When researchers stripped away extrinsic causes of death, things like car accidents, infectious diseases, and random environmental hazards, they found that the heritability of intrinsic human lifespan may be as high as 50% or more. This distinction is absolutely crucial. Think about it. In the 19th and early 20th centuries, a person with exceptional longevity genes might still die at 40 from tuberculosis or a factory accident. Their genetic potential was masked by a harsh environment. But in the modern era, where antibiotics, sanitation, and safety standards have removed many of these extrinsic threats, our genetic baseline is becoming the primary determinant of how long we live. We are essentially clearing the noise to finally hear the signal of our own DNA. Despite this high estimate of heritability, scientists faced a problem known as missing heritability. Genome-wide association studies, or GWAS, which scan the genomes of thousands of people, historically struggled to find the smoking gun genes for longevity. The answer to this puzzle likely lies in the distinction between common variants and rare variants. Most genetic studies focus on common changes that appear in at least 1 to 5% of the population. These variants usually have tiny effects. One might increase your lifespan by a few weeks, another might decrease it by a month. But the New England Centenarian Study and similar projects have found that exceptional longevity, living to 100, 105, or 110 plus, is often driven by rare, potent genetic variants. These are not small nudges, they are powerful shields. The study found that centenarians actually have just as many bad genes or disease risk alleles as the average person. The difference is that they possess a unique arsenal of protective genes that buffer them against the damage. This buffering effect becomes stronger the longer you live. The genetic profile of a semi-supercentenarian, someone aged 105 to 109, is significantly more distinct and protective than that of a mere centenarian. So what are these protective genes? While hundreds of genes play a role in aging, a select few have emerged as the titans of longevity research. Let's break down the usual suspects. If there is a single longevity gene, FOXO3 is the strongest candidate. Found on chromosome 6, this gene encodes a transcription factor, which is a protein that acts like a switch, turning other genes on or off. You can think of FOXO3 as the cellular equivalent of a crisis manager. When a cell is under stress, whether from a lack of food, heat shock, or oxidative damage, FOXO3 activates a suite of repair programs. It triggers autophagy, which is the cell's internal recycling system. It boosts antioxidant defenses, and it repairs damaged DNA. A specific variant of this gene, known as the G allele, has been consistently found in centenarians across diverse ethnic groups, from Japanese and German to American and Han Chinese populations. Having just one copy of this protective allele significantly increases the odds of reaching 100. Having two copies triples those odds. It effectively keeps the body's repair systems on high alert, ensuring that minor cellular damage is fixed before it can accumulate into catastrophic failure. Next, we have APOE, often called the double-edged sword. This gene is best known for its role in lipid metabolism and Alzheimer's disease. It comes in 3 common flavors or alleles, epsilon 2, epsilon 3, and epsilon 4. APOE epsilon 4 is the risk variant. Carrying one copy increases the risk of Alzheimer's by 3 to 4 times, and 2 copies can increase it by up to 15 times. It is also linked to higher cardiovascular risk. In studies of centenarians, the epsilon 4 allele is conspicuously absent. It is essentially an anti-longevity gene. Its presence acts as a barrier that few cross to reach extreme old age. On the flip side, APOE epsilon 2 is the longevity variant. It is relatively rare in the general population, but highly enriched in centenarians. It seems to protect against both Alzheimer's and heart disease, acting as a metabolic shield that preserves cognitive and cardiovascular health well into the 10th decade of life. Another key player is CETP, the cholesterol connection. The CETP gene regulates the transfer of cholesterol esters between lipoproteins. A specific mutation in this gene results in lower levels of the CETP protein and larger, fluffier HDL particles. That's the good cholesterol. This mutation was famously identified in a population of Ashkenazi Jewish centenarians by researchers at the Albert Einstein College of Medicine. These individuals had remarkably healthy lipid profiles and a significantly reduced risk of heart disease and stroke, even if they didn't always have the healthiest diets. This gene highlights the importance of cardiovascular health as a primary gatekeeper to longevity. Finally, among the titans we have SIRT1, the repairman. SIRT1 belongs to a family of proteins called sirtuins, often dubbed the genes of wisdom or guardian genes. SIRT1 is critical for DNA repair and genomic stability. It is the gene famously activated by resveratrol and calorie restriction. While the hype around drinking red wine for longevity has faded, the biology of SIRT1 remains solid. It helps maintain the integrity of the genome, preventing the accumulation of errors that define aging. To truly understand how these genes work, we must look at the machinery they control. Aging is not a single process, but a cascade of failures across multiple systems. One of the most famous mechanisms involves telomeres, the cellular fuse. Telomeres are the protective caps at the ends of our chromosomes, often compared to the plastic aglets on shoelaces that keep them from fraying. Every time a cell divides, its telomeres get a little shorter. When they become too short, the cell enters a state of senescence, a zombie state where it stops dividing, or it simply dies. For years, telomere length was touted as the ultimate biomarker of aging. While it is true that short telomeres are associated with mortality, the picture is complex. Recent consensus suggests that telomere length is less of a driver of aging and more of a limit on cellular replication. Interestingly, lifestyle factors can influence this fuse. A 2024 study on U.S. adults found that regular strength training was significantly associated with longer telomeres, suggesting that muscle tissue acts as a reservoir of systemic youth signals. However, telomere length has been surpassed in predictive power by a new generation of biomarkers, epigenetic clocks. While our DNA sequence, the hardware, remains fixed throughout our lives, the chemical tags that attach to it, the software, change as we age. This is epigenetics, specifically DNA methylation. Dr. Steve Horvath developed the first epigenetic clock, showing that by measuring methylation levels at specific sites on the genome, you could predict a person's chronological age with frightening accuracy. Newer clocks, like Grimmage, go a step further. They don't just tell you how old you are, they tell you how fast you are aging. Grimmage is currently the gold standard for mortality prediction, outperforming telomere length and traditional risk factors. It integrates data on smoking history, inflammation proteins, and methylation patterns to predict lifespan and health span. Crucially, these clocks have shown that biological aging is reversible. Interventions like diet, exercise, and emerging drug therapies have been shown to turn back the Grimmage clock in small clinical trials. We also cannot ignore the power plant problem, mitochondria. Mitochondria are the energy factories of our cells, and they have their own DNA, inherited exclusively from the mother. Over time, mitochondria accumulate damage and become less efficient, leaking toxic exhaust in the form of reactive oxygen species. This concept is known as the mitochondrial free radical theory of aging. Interestingly, genetic haplogroups, which are ancestral lineages of mitochondria, play a role here. Haplogroup J, common in Northern Europeans and Finns, is overrepresented in centenarians. It is hypothesized that this haplogroup produces slightly less energy and heat, but also fewer damaging free radicals, essentially running the engine cooler and longer. Similarly, haplogroup D4A is the Asian equivalent, strongly linked to longevity in Japanese centenarians, particularly in Okinawa. These findings suggest that a thrifty metabolism, one that prioritizes efficiency and low damage over maximum power output, may be a key to extreme endurance. Now let's discuss the pathways and paradoxes, specifically the trade-off between growth and repair. One of the most conserved longevity pathways in nature involves insulin and insulin-like growth factor 1, or IGF-1. In simple organisms like worms and fruit flies, reducing insulin signaling can double lifespan. The logic is evolutionary. When food is scarce, the organism shifts from growth mode to maintenance mode, prioritizing repair over reproduction. In humans, the story is more complex. High levels of IGF-1 are necessary for growth and muscle maintenance in youth. But in late life, high IGF-1 is linked to cancer and shorter lifespan. Conversely, centenarians often have naturally lower levels of IGF-1 signaling or mutations in the IGF-1R receptor gene. This creates a trade-off. The growth that makes us strong in our 20s may be the same force that ages us in our 80s. This is a classic example of antagonistic pleiotropy, genes that are good for us early in life but bad for us later. Closely related to this is the mTOR pathway. mTOR is a nutrient sensor. When you eat protein, mTOR turns on, telling the cell to grow and divide. When you fast, mTOR turns off, triggering autophagy, that cellular cleanup process we mentioned earlier. Chronic overactivation of mTOR, due to the constant overeating typical in modern Western diets, inhibits this cleanup process. This leads to the accumulation of cellular junk, such as misfolded proteins and damaged organelles, which clogs up the cell and leads to aging. Many longevity interventions, including the drug rapamycin, work by inhibiting mTOR, artificially tricking the body into a state of fasting and repair. The genetics of longevity are not just abstract data points. They are lived realities in specific enclaves known as blue zones. Take Okinawa, Japan. The Okinawan centenarian study has documented a population with remarkably low rates of heart disease and cancer. Genetic analysis shows a clustering of favorable FOXO3 variants and the mitochondrial haplogroup D4A. But their genetics also interact with a unique environment, a calorie-restricted, nutrient-dense diet, rich in purple sweet potatoes, and a strong social network known as MOI. Then there is Sardinia, Italy. In the mountainous Ogliastra region, there is a near 1-to-1 ratio of male to female centenarians. Elsewhere in the world, women outlive men 4-to-1. This population has been genetically isolated for thousands of years, enriching specific variants. Interestingly, they carry genetic markers often associated with inflammation. However, in their specific environmental context, characterized by high physical activity and a Mediterranean diet, these genes may boost immune response rather than causing chronic disease. This brings us to the future of longevity. We have moved from merely reading the code of life to having the tools to edit it. The future of longevity genetics is active intervention. One of the most promising frontiers is senolytics, drugs that selectively kill senescent cells. These zombie cells accumulate with age and secrete inflammatory chemicals that poison their neighbors. Early human trials with the leukemia drug dasatinib and the plant pigment quercetin have shown promise in clearing these cells and improving physical function in patients with pulmonary fibrosis. The goal is to periodically flush the body of these aged cells, reducing the inflammatory burden and allowing healthy tissues to regenerate. Then there is CRISPR and gene therapy. CRISPR technology allows us to edit genes with precision. In mice, researchers have already used CRISPR to silence the KAT7 gene, a driver of cellular senescence, resulting in extended lifespan. While editing the human germline to create designer babies remains ethically fraught and illegal in most places, somatic gene therapy in adults is on the horizon. Imagine a therapy that delivers an extra copy of FOXO3 or SIRT1 to your liver or heart cells via a viral vector, boosting your body's resilience as you age. Another target is PCSK9, a gene that regulates cholesterol. Editing it could permanently lower heart disease risk, mimicking the natural protection seen in some centenarians. Sometimes, however, the best anti-aging drugs are ones we already have. SGLT2 inhibitors, originally designed for diabetes, are showing remarkable off-target benefits. They reduce heart failure, protect kidneys, and in animal models, extend lifespan by mimicking some of the metabolic shifts of calorie restriction. In 2025, research began to solidify the link between these drugs and reduced markers of senescence in humans, suggesting they may be true geroprotectors. As we unlock the genetic keys to extending life, we must face profound ethical and societal horizons. If longevity therapies become available, will they be the preserve of the wealthy, creating a biological caste system where the rich live to 150 and the poor die at 75? Furthermore, what happens to the human species if we slow down the generational turnover? Death and replacement are the engines of evolution. By extending individual lives, we might inadvertently stagnate our cultural and biological evolution. Yet, the ethical imperative to reduce suffering is powerful. Aging is the primary risk factor for every major killer: cancer, heart disease, dementia. To treat aging is to treat the root cause of human suffering. In conclusion, the genetics of human longevity is a story of resilience. It is written in the rare variants of the supercentenarian, the methylation marks of the epigenetic clock, and the signaling pathways that balance growth with repair. We are learning that while we cannot change the hand of cards we were dealt at birth, we can change how we play them. Through lifestyle, pharmacological intervention, and potentially gene editing, we are moving toward a future where intrinsic aging is not an immutable decline, but a manageable condition. The 50% of our longevity that is genetic is no longer a fixed fate. It is a map, and we are just beginning to explore the territory. Thank you for listening to G Fun Facts Online.